Okay, to begin. So what we're going to do today is we're going to finish that story on the mitochondria. If you remember, I closed yesterday with asking you to think about how Lynn Margulis might have figured out that mitochondria were symbiotic organisms and had arrived from the capture event some time ago. And we'll get into that first because we're going to talk about the idea that mitochondria are characterized by a kind of prokaryotic set of nucleic acids. And we'll see that there are only 30-some genes in the mitochondrial genome, and that most of those are dedicated to making RNA and transfer RNAs and ribosomal RNAs that are unique to the mitochondria. And we'll talk a little bit about that. Then we'll spend a little bit of time on mitochondrial inheritance. Mitochondrial inheritance is all through the matrilineal line, and this has to do with the way the egg is fertilized and the survival of the mitochondria in the egg. And we'll talk a little bit about that and the implication it has for evolution and implication for understanding evolution. The next thing we're going to talk about is cytoskeleton and vesicle transport. Up to this point, we've emphasized all of the membrane-bound compartments, those compartments that have biological membranes surrounding them. And if you would imagine the cells simply being these membrane-bound organelles, you think of it as sort of like chicken soup with things floating about in the cytoplasm. It's not that way at all. The cytoplasm rather of the cell is highly organized. And it's highly organized by the cytoskeleton. So we're going to spend a little bit of time talking about the cytoskeleton, and then from the discussion of the cytoskeleton, we're going to talk about vesicle transport. We mentioned vesicles in passing yesterday. We're going to see that these cargo-carrying containers are going to be filled with proteins destined for secretion, or proteins that are destined to be incorporated into the plasma membrane, and then can be transported over vast distances by using the cytoskeleton. So we're going to first talk about the cytoskeleton and then spend a little bit of time talking about vesicle transport and the molecular motors responsible for vesicular transport. The cytoskeletal proteins are all protein polymers. And what I mean by that is we're going to have a particular protein. In the case of microtubules, we're going to polymerize tubulin into these great long molecules. Now, we're going to see as we move farther through this that for microtubules, these tubulin molecules can assemble, they can self-assemble end to end and side to side. And in so doing, they generate this tubule. And the tubule then will provide a substrate along which we can move vesicles using a particular motor, and we'll talk in some detail about that. The other major polymer we're going to talk about is actin, or an actin filament, sometimes known as F-actin or filamentous actin. And the actin filaments are polymers of these much smaller molecules of globular actin. The globular actin has a molecular weight of about 40,000, and it can self-assemble in these actin filaments, and we'll talk a little bit about that. We're going to spend most of our time on microtubules and actin filaments because these are the filaments, these are cytoskeletal components that support transport. The third set of polymers that we see in the cytoskeleton are things known as intermediate filaments. We're going to run into intermediate filaments again when we talk about skin because they're a predominant kind of structure in keratinocytes. We're going to see intermediate filaments also in neurons called neuronal filaments. And these intermediate filaments are basically just structures that provide structure to the cell. They're not supporting transport, so we're not going to spend much time on them. Then we're going to spend a little bit of time about the unique characteristics of how we can organize the microtubules and actin so that we can have transport on them. We'll see that microtubule organization, as I mentioned previously, is that it's a polymer of tubulin, but the tubulins then will assemble side to side and end to end. And what this means then is as we assemble these structures, there's a plus end and a minus end. The plus end simply means the addition end. It's where we add tubulin monomer on. The minus end is where we take the tubulin off. And these polymers are characterized by this ability to what they call treadmill, where we can add at one end and take them off at the other. Now, we're going to be talking almost entirely about microtubules that have been stabilized, particularly in neurons, but the rules are pretty much still the same. That is, we have these giant polymers of molecules called tubulin that will extend for, maybe as much as 100 micrometers. So they're enormous. The other structure that I'm going to talk about is filamentous actin, and again, this is going to be polymerized from globular actin. We're going to think about the globular actin as sort of like a pop bead, and we'll be adding these G-actins to make a polymer. Now, if you get into a biochemistry course and you get into the details of this, you'll find that the regulation of polymerizing G-actin into filamentous actin, there are whole host of proteins involved with severing the actins and polymerizing them and controlling the polymerization and getting them to branch. We're not going to get into that. I just want you to recognize that we can take globular actin and plug it end to end and make a polymer out of it. And that, like the tubulin, the globular actin will assemble in a particular way. So there's a plus end to the filamentous actin and a minus end. And we're again use that endedness for transport. When we're talking about microtubules, and this is an important idea that we'll spend some time emphasizing, is that we have a plus end directed motor. The plus end directed motor will drag vesicles along the microtubule toward the plus end of the microtubule. And we'll discuss how this was discovered and discuss its importance. But these microtubules can support transport in both directions. And so there's another motor called dynein, which is a minus end directed motor. So we're going to lay out these giant filaments that go long distances. And now we can have motors which can move traffic in both directions along these substrates of the microtubules. One being kinesin, being a plus end directed motor, and dynein being the minus end directed motor. We're going to lay this story out in neurons, and this presages what we're going to talk about a little bit later in the course in neurobiology. But this segment here, I like to use the neurons because this is where where kinesins were first discovered. They play an important role, and we can see this is where they first discovered that microtubules are oriented, at least in the axon, in a particular way. And we'll talk a little bit about that and talk about how we can move proteins over long distances using kinesin, and that the kinesin uses ATP as an energy source. That major concept I want you to hold on to when we get to this part of the discussion is that these enzymes, whether we're talking about kinesin as the plus end directed motor or dynein as the minus end directed motor, or whether we're talking about myosin on actin, all of these molecules use ATP as a source of chemical energy. And what they can do, what their trick is to convert the energy in a chemical, ATP, and convert that chemical energy into mechanical movement by changing the conformation of the molecule. And we'll talk a little bit about how this happens. It's pretty well worked out, particularly for kinesin and for myosin. And the issues are still somewhat unclear on dynein. And we'll touch a little bit on that. But let me remind you where we were last time, and we were finishing the discussion of mitochondria. And we have described the importance of mitochondria to the cell, that they are the site where we generate most of the cell's ATP. We have mentioned that they look a little like this. And what distinguished them from most of the other organelles is they had a double membrane. And that Lynn Margolis, one of these early electron microscopists in the 1950s and 1960s, noticed that these structures had double membranes, and she said, well, maybe they're related to bacteria. And so she wanted to test that hypothesis. And so what she did is she grew up cells in culture. She grew up millions and millions and millions of cells in culture, scraped those cells off of the dish, and then asked the question, what kind of nucleic acids could she find in mitochondria? Because if mitochondria are a kind of modified bacterium, then they should have prokaryotic nucleic acids. And sure enough, when she took these millions and millions of cells, spun them down and isolated, resembled prokaryotic nucleic acids. Just a reminder that this term nucleic acids includes both the deoxyribonucleic acid, DNA, and the ribonucleic acid. And we'll get into that a little later, probably today and maybe tomorrow, probably tomorrow. But this then was a pretty good indication that these mitochondria were related to bacteria. Now, I'll give you some numbers. Don't remember these numbers, but I think they're interesting. It turns out that there are 37 genes in the mitochondrial genome. Of these 37 genes, 13 to 14 make proteins, are responsible for proteins. There are 2 that are responsible for ribosomal RNA, and 22 responsible for transfer RNA. Now, I find this sort of interesting because what's been conserved in the mitochondria are predominantly these genes that are responsible for making transfer RNA and ribosomal RNA, so because the protein synthesis machinery has been retained here from the old bacterial genome. Now, I don't want to leave you with the idea that these mitochondria only use these 37 genes. In fact, many of the proteins that are functioning in the mitochondria are imported from the cytoplasm. There are genes in the autosomal genome in the nucleus which will make proteins that are important for mitochondrial function, and they have to be imported into the mitochondria. So over the last 1.2 billion years or so, these symbionts, these endosymbionts, the mitochondria, have evolved with the cell itself. And so there are autosomal genes where this is the nucleus. There are genes in the nucleus that generate proteins destined for the mitochondria. Truth in advertising. Yesterday, when we divided our protein types into these enormous categories, we said some were cytoplasmic proteins and some were made on the rough endoplasmic reticulum. Well, this is an exception to that in that these are cytoplasmic proteins, but they have a special import system that will import those proteins into the mitochondria. Take a cell biology course, you'll learn more about that. We're going to pass right on by it. But recognize that although the nucleic acids in the mitochondria are typically prokaryotic nucleic acids, they still require the importer proteins to allow for their function. So are there any questions up to there? Well, if not, let me spend a little bit of time on the right-hand board because I think this is really interesting in terms of mitochondrial inheritance. Now, the story begins, as all life begins, with the ovum. This is the egg. Now, in the ovum, in humans, there are 23 chromosomes. It's said to be haploid. For this egg or ovum to mature into an embryo and mature into a new individual, it requires another set of genes. And those other set of genes, of course, arrive I'm going to put one of these in here. They arrive carried in the nucleus of a sperm. You'll learn much more about this at the end of the course, and Dr. Bauma comes in and lectures on reproduction. But what will happen is that this sperm has been propelled in the final stages toward the egg by the flagellum. The flagellum requires ATP, and the ATP is generated by a mitochondrion. In the sperm. But when the plasma membrane of the head of the sperm fuses with the plasma membrane of the ovum, and the nucleus of the sperm enters the cytoplasm of the egg, this mitochondrion is lost. That's to say that none of the mitochondria in the sperm are contributed to the next organism. That all of the mitochondria that are going to be in the sperm, all the mitochondria that are going to be in the developing organism are in the ovum. So this is the maternal mitochondrion. This has implications because it means that the mitochondria that you have in your body, you got from your mother. And she got them from her mother. And she got them from her mother all the way back until probably 200,000 years ago or so, when we see the first modern humans arising in Africa. It turns out that mitochondrial DNA in Neanderthals and Denisovanians is different than the mitochondrial DNA of humans. But all of the mitochondrial DNA of humans comes through the matrilineal line. So that means we can look at these genes. We can take the genes, we can take the DNA in the genes of the mitochondria and begin to look at how they've changed over time. And so by looking at mutation rates and having some idea, a vague idea of how these mutation rates are going to occur, we can then look at individuals living in particular parts of the world and ask the question, how long has this group of people been in that region? And one of the things we find is that when we look at the ancestral regions where human beings first evolved, that is back in Central Africa, that's where we find the greatest variability in the genomes of the mitochondria. As people moved out of Africa and over the last 30 or 40,000 years began to occupy Asia and Europe, we find that those groups now will have more similar mitochondrial DNA because they've lived together for the most recently. And so when you look at European groups, you'll see mitochondrial DNA, which is similar among the different groups of people. And this has helped track the movement of humans through Europe. And there's been a really big argument about what's happened to the hunter-gatherer populations in Northern Europe when the agricultural folks moved in from the Anatolian Peninsula. And they've been able to track some of this through the mitochondrial DNA. And it looks like there was a massive displacement. It looks like most Northern Europeans are related to, ancestrally related to, the farmers, the farmers who came in from areas around Turkey. And there aren't many people who are related to the old hunter-gatherers that were there 9,000 years ago. So it looks like there was a massive displacement of the hunter-gatherer groups that were in Northern Europe, and that displacement was by those people who came out of the Anatolian Peninsula, which is now Turkey. That's where most of the farming came from in the Fertile Crescent and the Anatolian Peninsula. And it looks like they displaced the hunter-gatherers. And much of that evidence comes from understanding the matrilineal lineage of the mitochondrial DNA. So any questions about that?
Could you real quick just touch on the fact that the autosomal DNA and the mitochondrial DNA are different from different places?
Okay, an important question here has arisen about the distinction between what I can label here as autosomal DNA. The autosomal DNA is the DNA that's sequestered to the nucleus. It's the autosomal DNA, which you have inherited 23 chromosomes from your father, from the sperm, and 23 chromosomes from your mother, from the ovum. And that genetic material always resides in the nucleus. There is this tiny bit compared to the 20,000 or so genes in the nucleus of the autosomal DNA. There are about 30 genes out here, what did I say, 32 genes out here that as part of the prokaryotic DNA, which are confined to the mitochondria. So these are in different places. The autosomal DNA always resides in the nucleus. The prokaryotic DNA, which is a remnant of this being a bacteria 2 billion and a half years ago, 1.2 billion years ago, that means this DNA and RNA is only in the mitochondria. So keep those separate. Keep those separate. The autosomal DNA is always in the nucleus, and this is a very special subset of DNA that shows that these structures were captured a long time ago. As an aside, to emphasize this relationship to bacteria, the motor, I'm going to do it this way, that there are motors in wild bacteria that drive the flagella. It's a different flagellum we see in the sperm, but they drive the flagella to move the bacterium. And people have been studying these motors for a long time, and when they looked at the structures inside the mitochondrion that was making the structures that were making ATP, they were almost identical structures to the flagellar rotors. They don't have flagella on it anymore, but they still use a hydrogen ion gradient. The hydrogen ion gradient drives this rotor, and by driving the rotor allows ATP to be generated, and that's all done by the hydrogen ion gradient and the remnants of the old flagellar motors. Any questions up to there? Okay. Well, with that, if there are no more questions, let me show you this picture, and we can sort of briefly review the different parts of the cell we spent the last 2 days talking about. And I won't spend a lot of time on this. I just wanted to point out a couple of things and give you a sense of what we've been drawing. Here's the nucleus. This is a specialized region of the nucleus called the nucleolus. We didn't spend much time on it. Remember, the nucleus contains all of the DNA. This is a bag of DNA. The DNA has been organized by histones. Those DNA binding proteins will organize the chromosomes inside the nucleus. But there are some regions which you can see they're splayed open, and these regions are the nucleolus. And these are the several different chromosomes contribute genes in this region, and we use those genes to make the ribosomal RNA. And there's so much ribosomal RNA being generated in every nucleus that we have a specialized portion of the DNA, the nuclear DNA, which is called the nucleolus. These sort of fluorescent green spots are meant to represent the nuclear pores, which, recall, will regulate movement from the nucleus into the cytoplasm, and to a lesser degree from the cytoplasm back into the nucleus. There are these, this is a membrane-bound structure, and it doesn't show it here, but this membrane here is continuous with the membrane of the endoplasmic reticulum. The endoplasmic reticulum here is shown in light blue. The rough endoplasmic reticulum is shown here, where we have these beads on the endoplasmic reticulum. These orange structures are meant to be the mitochondria. This series of layered structures is meant to be the Golgi apparatus. And we see that vesicles will bud off from the endoplasmic reticulum carrying proteins. Could be lysosomal proteins, could be proteins destined for export. It could be transmembrane proteins. These proteins then will be passed to the cis stack of the Golgi apparatus, processed in here, and out of the trans stack will come the finely processed proteins packaged into vesicles that are either destined to go to the plasma membrane, where they'll release their contents to the outside world by exocytosis, or they're destined to go to the lysosomes. Here, these purple structures, they're destined to go to the lysosomes and deliver that subset of proteins, the lysosomal proteins, into the lysosome. And we discussed yesterday how endocytosis in specialized cells can bring bacteria in from the outside, deliver them to the lysosome, where they'll be destroyed. In most cells, these lysosomes are functioning day to day to break down worn out proteins. And then down here's the cytoskeleton, which we're going to head toward next. Any questions on that? In that case, let's move on to the cytoskeleton. And again, if you looked at that image, you could imagine all of those organelles floating about in the cytoplasm as though they were free and untethered to one another. But that's not true, because what we're going to see is that one of the major constituents of eukaryotic cells are going to be cytoplasmic proteins. Cytoplasmic proteins that form the cytoskeleton. Now, there are a lot of components to the cytoskeleton, but we're going to stick to the basic structures. We're going to say the cytoskeleton is composed of one, microtubules, two, something known as filamentous actin, and three, intermediate filament. Protein. These molecules, these cytoskeletal elements, were described early on just simply from their structure. And the filamentous actin were described as microfilaments. Sometimes they were referred to as 7 nanometer filaments. In some cases, the microtubules are referred to as 25 nanometer filaments. And by default, the intermediate filaments, which were in the neighborhood of about 11 nanometers, got their name simply from the structure. And to this day, this sticks. These intermediate filaments we call intermediate because of their intermediate size between the microfilaments, which were only discovered later to be actin, and the much larger microtubules, which were the 25 nanometer filaments. What all of these have in common, proteins, are polymers of smaller proteins. We'll see that the microtubules are polymers of a protein called tubulin. Now, tubulin itself is a dimer with an alpha and a beta. The filamentous actin is a polymer of globular actin. This globular actin is sometimes referred to as G-actin. And then finally here, may as well put it in orange, the intermediate filaments. Are polymers of intermediate filament protein. These intermediate filament proteins may be things like keratin in keratinocytes, of skin, or they may be neurofilaments. In neurons. There are also intermediate filaments in glial cells. We're not going to get into those. But I'm not even going to spend any much more time on the intermediate filaments because they're not important to our discussion in that these are only structural proteins. They don't really support transport. And what I want to talk about today is how these molecules, the polymers of microtubules made of tubulin and polymers of actin made of actin can support the movement of vesicles. Because once you become a eukaryotic cell and you're producing proteins and packaging them in vesicles, you have to move them from place to place. We saw that to some extent in moving these vesicles from the endoplasmic reticulum to the Golgi apparatus, and we saw again that we had to transport the vesicles either to the lysosomal system or to the plasma membrane. And so that means there must be some form of transport. And what I want to do is emphasize the role of these cytoskeletal proteins, the microtubules and the filamentous actin in transport. And since the molecules of intermediate filament protein don't do much in the way of transport, we're going to leave them behind for the time being. So what I want to do in this panel is I'm going to erase the structure of the cell and begin to think about how the microtubules can polymerize from individual protein molecules and the implications of this polymerization for the function of the final microtubule. And so what we're going to do is imagine in our mind's eye, in some of my crude drawings, how we can take the tubulin molecules and assemble them end to end and side to side to make a microtubule. And how this assembly, the self-assembly of the tubulin molecules into the much larger microtubule structure supplies a kind of endedness to the molecule, which then can be used by motors to move in one direction or the other. But first, let's simply talk about the polymerization. Polymerization of tubulin into microtubules. Now remember, we started out with the idea we had these individual tubulin molecules, which are themselves dimers. They look a little bit like dumbbells or a little bit like figure eights, where there's an alpha and a beta. Well, one of the beauties of these molecules is when you get the concentrations of tubulins high enough, they'll start to self-assemble. And so this alpha beta will self-assemble end to end with another alpha beta, which will self-assemble end to end to another alpha beta, and so forth. Alpha beta. Now, if that's all there was to the story, you'd have one single line of tubulins that are assembled end to end. But we can say here that tubulin molecules assemble end to end. But in addition to assembling end to end, they also assemble side to side. So that this alpha and beta will assemble side to side to another alpha beta. Again, they'll be, in this strand, they'll be organized end to end. Alpha, beta, alpha, beta, alpha, beta, and so forth. But now side to side. Now, if you carry this out to its logical end, and unlike my drawings, these alpha betas are all identical. Now, if you would carry this out to its logical end, we end up with a sheet, and that's not what happens. When these alpha-beta tubulins assemble side to side, each one assembles in such a way that it adds a little bit of curvature. It adds 1/13 of 360 degrees. So when we look at these in cross-section, so if we take this series of tubular molecules laid out end to end, now we cut it here and rotate it 90 degrees. So imagine now we take this, my arm is this series of tubular molecules that are assembled end to end and side to side. Now we're going to turn it on end and look down the axis of that tubule. When we do that, we see that these tubulins are arranged in a circle. And they're arranged in a circle always, so there are 13 of them. So there are 13 tubulins in a circle to construct the microtubule. So this is just an extension of the idea that they're assembled side to side, they're assembled end to end, and when they assemble side to side, there's a certain curvature giving this tubular structure. This diameter of this tube is in the neighborhood of about 24 to 25 nanometers. So not very big. I mean, these are molecular scales, but you remember that a typical phospholipid bilayer is 6 or 7 nanometers wide. So these are, 3, 4, 5 times the width of the phospholipid bilayer, but these are inside the cell. The other important aspect of the assembly of tubulins is such that tubulin adds at one end of the microtubule and comes off at the other. Well, the real implications for this seems like a simple idea, but the real implications for this, because the microtubules will be formed by the addition alpha beta at this end, so this is the addition end, and they'll come off at this end, this is the subtraction end. We abbreviate this to say that this is the minus end, and this addition end is the plus end. But now the orientation of the alpha betas and the way these alpha betas will now add together, the way they'll self-assemble, gives a kind of endedness to the molecule. It gives a plus end and a minus end. So if we were to simply draw this out simply and say this line is a microtubule, there's a minus end to the microtubule and a plus end. This gives an orientation to the microtubule. It gives an orientation to the polymer. It tells us an endedness to this particular molecule, and that's going to be important here in a moment. And it's at this end where we add tubulin. Try again. It's at that end where we add tubulin. And this end, tubulin, comes off. Well, of course, if you took this to its logical conclusion, you'd say that these microtubules inside a cell are constantly churning, where we're adding tubulin to one end and coming off at the other. And in some cases, that's true. But in the case I'm going to tell you about now, where we have microtubules in neurons, these microtubules and neurons are stabilized. So that they're pretty much fixed. So the next thing we're going to do is we're going to lay out these microtubules in a cell. But before we get to that, because we're talking about polymeric molecules, polymeric proteins, let's go to the right-hand panel and discuss how we can do something very similar with globular actin. So remember we said the globular actin, G-actin, can be assembled into a filament called filamentous actin. And the rules are very much the same. It's a little bit different if you get down into the cell biology of it and the molecular biology of this story. They're a little different, but what I want to tell you is that we can now take a monomer of actin and have it assemble end to end, and by assembling end to end, it can generate a filament. So we'll take this panel and describe polymerizing filamentous actin. From globular actin. Now, the fact of the matter is that when we polymerize these molecules, there are a whole host of other proteins that are involved in this polymerization. There are a whole host of them that are involved in cutting them. We're not going to get into that. If you take a cell biology course, you'll learn about those. But what I want to emphasize here is that we're simply going to polymerize a filament of actin from globular actin. And I'm going to show these globular actins simply as circles. Each G-actin has a molecular weight of about 40,000. And they all assemble end to end. Now, typically, when we look at one of these filaments, these actins have assembled in such a way that they're offset from one another. But just as we saw with the formation of microtubules, when we form these actin filaments, where this is an actin filament, polymerized from globular actin. Sometimes known as G-actin. We see that we are plugging these together like pop beads on a string, and that again, there's going to be an end to which G-actins adds, and an end from which G-actins come off. As we saw with microtubules, this now provides a polarity and endedness, so that this is the plus end to which we add on, and this is the minus end from which the actins fall off. So now we have this organization of a filamentous actin with a plus end and a minus end. Well, that endedness becomes important because we're going to see, well, it looks like tomorrow, we're going to see that the motors are going to move in one direction or the other along these molecules. But on the left-hand panel, let me try to introduce the microtubules into a cell and introduce the microtubules into a particular kind of cell, an unusual cell called the neuron. And the reason I tell this story is twofold. The functions of the microtubules in their role of vesicle transport was discovered in neurons. And it's a good place to think about it because neurons are these highly asymmetric cells. They are extremely asymmetric, and yet all of the protein synthesis in a neuron occurs in a circumscribed region. It occurs in the cell body. That means these proteins, particularly the ones that have been generated on the rough endoplasmic reticulum and packaged into vesicles, have to be moved from their site of synthesis to the target where they're going to be used. So I want to think a little bit about the organization of neurons and then begin talking about how vesicles and molecular motors may work. So this is going to be the neuron as an asymmetric cell. I'm going to draw this crudely, and we'll label this briefly. And we're going to get into way more detail as we talk about neurons. But I'd like to bring this up at this point because it tells a nice story about the microtubules. This part of the neuron is said to be the cell body. And when we look carefully at the cell body, we see that it obeys the rules we talked about for ordinary cells as the nucleus. And just as we saw in other cells, around the nucleus will be this membrane brown bag structure of the endoplasmic reticulum. And then, of course, included within that will be the Golgi apparatus that we discussed yesterday, and the generation of vesicles. These are vesicles containing proteins, transmembrane proteins, and proteins destined for secretion. But now arises the logistical problem of the neuron. All protein synthesis occurs in the cell body. But yet, all along this great expanse of an axon, which in some cases, in extreme cases, may be greater than a meter long, and down here at the axon terminal, that may be more than a meter away from the site of protein synthesis. What this tells us is that the proteins that are made here and put into vesicles need to be distributed in some way along this great long structure and arrive at their output regions here where we can use the proteins. And it was a mystery about how this was done for a long, long time. But one of the first things they discovered when they began studying exoplasmic transport is that inside these axons are these great long structures, which we've just discussed. These are called microtubules. Now, where the total distance here may be a meter, these microtubules can sometimes, if they're big, be in terms of 100 micrometers, 0.1 millimeters, which seems like a short distance, but when you consider that each one of these tubulin molecules here is about 8 nanometers, these 8 nanometer individual proteins extending for 100 micrometers, these have a lot of tubulins in them. The final point I want to make here is that when they laid out these microtubules and began to explore how they were oriented, they were always in the axon oriented in one way, so that this is the plus end and this is the minus end. And every one of the microtubules they looked at was always oriented in the same fashion, so that the minus ends were closest to the cell body and the nucleus, the plus ends were closest to the terminal. This orientation, this endedness we'll see tomorrow, is important because it provides the substrate and the endedness for the molecular motors. And tomorrow we'll start talking about molecular motors moving vesicles along microtubules, and molecular motors in the end moving vesicles along actin filaments. So, any questions to there? Okay, well, let's wind it up then. Thanks.